Hello everyone and welcome to the 29th Annual GW Leeper Memorial Lecture. Thank you for joining us. My name is Professor Brian Lurie and I am the head of the School of Agriculture and Food at the University of Melbourne. On behalf of the school, the University and Soil Science Australia, welcome. Before we begin, I wish to pay my respects to the elders, past, present and emerging, of the traditional owners of the lands from which we are joining for this lecture. In my case, those are the Wurundjeri people of the Kulin Nation. As this is the first Leeper lecture to be delivered online, I'd also like to thank everyone who is joining us for the first time, particularly those of you from regional Victoria or interstate. This annual lecture in soil science was established in the name of Professor Geoffrey Winthrop Leeper, and has been sponsored by the University of Melbourne and the Victorian branch of Soil Science Australia since 1992. Professor Leeper spent most of his career at the University of Melbourne. He graduated here with a Bachelor of Science and a Master of Science in Organic Chemistry. He lectured at the Universities of Adelaide and Aberdeen before returning to the University of Melbourne in 1930 as a research scholar in chemistry. His research interests were in manganese and other trace elements in soil, as well as soil classification. He became the chair of agricultural chemistry in 1934 and acted as the dean of the faculty in 1939, 1944 and 1945. Professor Leeper was known for his contributions to soil science and chemistry, his insistence on academic rigour and accuracy, and his appreciation of plain language and rational thinking. Each year, the Leeper Lecture is delivered in that spirit by a distinguished soil scientist. This year's Leeper Lecturer is Caixian Tang, Professor in the Department of Animal, Plant and Soil Sciences and the Centre for Agribioscience at La Trobe University. Professor Tang completed his tertiary studies in agricultural science at Zhejiang Agricultural University in China. He gained his PhD from the University of Western Australia in 1992, and following several years in research, he joined La Trobe University in 2003. He has been recognised through several awards for his teaching and research training, including the inaugural La Trobe University Award for Excellence in Research Supervision last year. Professor Tang's research has focused on the mechanisms and management of soil acidification, the role of organic amendments in soil pH change and the amelioration of soil acidity, the effects of elevated carbon dioxide on soil carbon and rhizosphere biochemistry and nutrient acquisition. His lecture today, titled "The Role of pH in Soil Organic Matter Dynamics," will draw on much of that research. Please welcome Professor Caixian Tang.
Thank you very much, Professor Lurie, for your nice introduction. I'm very honored and humbled being nominated as a speaker of this year's Leeper Lecture. And thank you very much for the nomination. Today, I will share with you the research work my team did in the last 10 years or so about the impact of soil pH on the dynamics of soil organic matter in acid soils. As many of you are aware, that soil acidity is a major limiting factor affecting the global food production. So this slide shows that the distribution of acid soils internationally, as indicated by red color. It is estimated up to 50% of arable lands are acidic or potentially acidic. In Australia, there are about 77 to 90 million hectares of agricultural lands are acidic. Among these, 23 million hectares have the subsoil acidity problem, which is difficult to ameliorate, is below the pod layer. So the major factors limiting the crop production in acid soils include the toxicity of aluminum ions, manganese ions, and hydrogen ions, especially for nitrogen-fixing legumes, as well as the deficiency of the nutrients such as phosphorus, calcium, and potassium. So the surface application of lime is the most common practice to ameliorate the soil acidity. However, we are not fully understand whether the amelioration of soil acidity favors the carbon sequestration, how pH change affects the decomposition of soil organic matter. So in this lecture, firstly, I will talk about the short and long-term effects of lime application on the concentration, chemical composition, and stability of soil organic matter. I will then talk about the effects of crop residues on soil pH in respect of the crop residue type effect and initial soil pH effect. Finally, I will talk about the rhizosphere effect on the soil organic matter decomposition, includes the effect of plant species and effect of rhizosphere pH. So rhizosphere is a very small region between surface of the roots and the soil. It's generally about 1 millimeter region, and their property is totally different from bulk soils. Now, application of lime to the acidic soil obviously will increase the pH, increase the availability of calcium ions, but decrease the bioavailability of aluminum ions and changes the nutrient availability as well. As a result, lime application improves crop production and then increase the primary carbon inputs, which favors the organic matter accumulation. Lime application also can affect the soil structure, therefore can affect the physical protection of organic matter in the soil. Some studies, previous studies said that liming can improve the soil structure, but some other studies showing that lime can deteriorate the soil structure. Increased pH result from lime application can increase the solubility of soil organic matter. They can increase the microbial activity. As a result, they will increase the decomposition of soil organic matter. This is not good for the soil organic matter accumulation. It is not well known how the lime application and associated change of soil properties affect the microbial composition, diversity, and functions on the soil organic matter decomposition. So the net effect of lime application on soil organic matter really depends on the relative contribution of these processes. So now I will talk about the short-term effects of lime application on soil organic matter decomposition. And we applied C13 labeled calcium carbonate to the various depths of the soil columns. By using C13 labeled lime, we are able to separate the CO2 comes from the organic matter decomposition and the CO2 from the lime dissolution. Okay, so from here, from the slide, we can see that increased lime application to the soil can increase the decomposition of soil organic matter, as indicated the amount of CO2 release increase, but in the first 3 days. This is a similar story in the soil Chromosol, which have a higher organic matter content, and Tenosol, which have a lower organic matter content. However, this increased organic matter decomposition diminished after 3 days of incubation. After 3 months of incubation, you can see the lime application or increased lime application rate increased the soil pH very well in the applied layer. However, increasing the lime rate did not affect the CO2 released from soil organic carbon decomposition. As indicated, there are no significant effects for both soils. Although that's the concentration of the extractable organic carbon and sometimes the microbial biomass carbon are variably increased by lime application. So now we also looked at the long-term effects of liming on the soil organic matter. So we cracked the soil from the top 10 centimeter layers in the various long-term field trials with lime history of 5 to 34 years. So 3 of these trials are in the Western Australia, and 2 trials are in Victoria. Okay, so this slide shows the concentration of the total organic carbon, particulate organic carbon, humus organic carbon, and resistant organic carbon as affected by lime application or lime histories in the soil collected from Wongan Hill in Western Australia, the medium rainfall area. Okay, so the lime was initially applied 30 years ago, and some plots was relimed 15 years ago before the soil sampling. So from here, you can see the general trends that increase the lime application rate, decrease the concentration of the total organic carbon, except for the soil relimed 15 years ago. The same pattern or the trend followed for the concentration of particulate organic carbon and humus organic carbon. But the lime history did not affect the concentration of resistant organic carbon at all. So this slide shows the results in the soil collected from the Calabarin in Western Australia. So the lime was applied 5 years ago before the soil sampling, and you can see the general trends are very similar compared to the Wongan Hill side. Does this slide shows the concentration of the total organic carbon and the concentration of the carbon fractions in the soil collected from the Bandura Latrobe University farm. Okay, so the lime was applied 34 years ago before the soil sampling. So I really would like to thank Dr. Nick Urin, who established this lime trial before I came to Latrobe. So this site, there are minimal input of the carbon into the soil because there are very little crop growing or the crop was properly managed. So from here, you can see the story or the trend of the lime application the effect of lime application on the carbon side is very similar. So if we look at the soil from the younger lime plot, that means lime applied 6 years before the soil sampling. We generally see the same story. So it looks like the story is very consistent for the soil from the Western Australia and from the Victoria. So we use the same soil collected from the field trials and to conduct the NMR analysis. That means use the nuclear magnetic resonance spectroscopy. Okay? So with the NMR analysis, we are able to provide the information about the carbon composition of soil organic matter in the soil as affected by lime history. So this slide shows the spectrum of the soil from Wongan Hills, okay, 30 years after initial application of lime. And the red line or red spectrum indicates the soil of zero lime, the control soil. And then there are various soils and generates various kind of NMR spectrum. So if we look at the ratio of the RQ carbon to all RQ carbon, and we found that increased lime application either did not affect this ratio or have the trend to decrease this ratio. So this ratio generates the index of the humidification of the soil organic carbon in the soil, or an indication of the soil organic matter stability in the soil. So these are the spectra of the soils collected from herbarium 5 years after initial application. And you can see same story. This ratio, the RQ to ORQ carbon ratio, it was not affected by lime application or tend to decrease by the increase in lime application rate. So these are the spectrum of the soils collected from lime plots at the university campus. Okay, and here you can see increase the lime application rate, decrease the RQ to ORQ carbon ratio. So it indicates that the lime soil may not necessarily to be stable in terms of the organic carbon in the soil. Similar story for the soils from the younger lime plot, 6 years after initial application of lime before the soil sampling. Okay, so from these slides, we indicate that lime history did not appear to increase the carbon accumulation in the topsoil or improve the carbon stability in the topsoil. Then we collect the soil from the old lab plot on the Latrobe campus. And one is the soil with initial pH 4.7, another soil with the initial pH of 6.6. Then we adding the C13 labeled glucose and C13 labeled lignocellulose into the soil, and half of the soil we add the ammonium nitrate as a nitrogen source. So if we use C13 labeled carbon substrate, we are able to separate the CO2 from the carbon substrate and the carbon CO2 from the decomposition of organic matter. So from here, you can see that the carbon substrate addition increases the microbial biomass carbon while the nitrogen addition decreases the microbial biomass carbon. And if we look at the priming effect, that means additional decomposition of organic matter in the soil induced by addition of this carbon substrate. So that is additional compared to the control soil, okay? So we call it the priming effect. So from here, we can see that addition of these two carbon substrates increase the priming effect or accelerate the soil organic matter decomposition. This decomposition or acceleration of decomposition is higher in the soil with high initial pH and higher with the glucose. And addition of the nitrogen decrease the priming effect, probably due to the decrease of the pH and or the increased nitrogen availability, which can minimize microbial nitrogen mining to minimize carbon decomposition. So now we also use soil collected from long-term field trials in Western Australia. With and without lime history, we can see the pH varies from the increased by 0.6 units to 0.9 units. Then this time, we add in the C13 labeled wheat residue. Okay, so previous slides we used glucose and lignocellulose, which represent the soluble and insoluble organic compounds of the crop residue. So this time we add in the C13 labeled wheat residue. So from here we can see that the lime history increase the residue decomposition. And lime history also increase the priming effect. That means acceleration of the soil organic matter decomposition induced by the residue addition. And you can see the figure here. So now we also analyze roughly about the microbial composition. So from here, you can see the RQ gene abundance was increased by liming history, as well as the residue addition. In contrast, bacteria gene abundance was not affected by liming history, nor by the crop residue addition. In comparison, fungal gene abundance was increased by liming history or increased the pH, but not, or was not affected by liming history, but increased by residue addition, as shown in this table. We also quantified some carbon cycle related genes, functional genes. So from here, you can see the abundance of cellulose-decomposition genes was not affected by liming history, but enhanced by the residue addition. In comparison, the abundance of the lignin-decomposing gene, LMCO, was enhanced by liming history, as well as the residue addition. So we are not sure whether that indicates that liming history or lime application favors the decomposition of recalcitrant carbon in the soil. So in this experiment, we collect the soil from the lime plots with various lime application rates. So we generate the soils with a wide range of initial pH from 4.7 to 7.4. Okay. Then this time, we incubate the soil with the C13 labeled wheat stubble and field P residues. So from here, you can see the top two panels are the priming effect or the decomposition of the soil organic carbon induced by the residue addition as a function of the soil pH on x-axis. The bottom two panels are the residue decomposition. Okay, so clearly we can see that the optimal pH for the acceleration of the soil organic carbon decomposition is about 6.5. In comparison, the optimal pH of residue decomposition is at or was at the pH 7.5 or even higher because we still see the linear increase. So now we talked about that soil pH can affect the decomposition of crop residues. And at the same time, addition of crop residues per se can affect the soil pH, okay? Which in turn affects the decomposition of indigenous soil organic matter, especially in acid soils. So in the second part of my talk, I will discuss the effects of crop residue on soil pH. So the effect of nitrogen cycle on soil pH due to the crop residue addition is very well known. When we harvest crop, the crop residue returned back to the soil becomes the source of the soil organic matter. So during the decomposition of soil organic matter, the organic nitrogen will convert to the ammonium through the ammonification process. So this process is hydrogen ion consuming process, which increases pH. If the ammonium produces ammonium being immobilized by microorganisms or convert to the nitrate through the nitrification process, these two processes are the hydrogen ion producing process, which increases soil pH. If the nitrate been immobilized by the microorganisms or taken up by the crop plants or convert to the gases through the denitrification, all these three processes are hydrogen ion consuming process, which increases soil pH. So that is very well known. However, the effect of carbon cycle on soil pH upon the crop residue addition is not very well known and is more complicated. So from our work, and we showed that the addition of the crop residue and the subsequent decomposition and transformation to organic matter can generally increase the soil pH, depending on the residue type. And in the first part, I discussed that addition of crop residue can stimulate the decomposition of soil organic matter, so termed as a priming effect. So the priming effect can lead to the pH increase to a certain extent from our studies. As I also mentioned, that crop residues have the soluble components, have an insoluble fraction, and organic compounds can be anion form as well as acid form. So we use the concentration of excess cations as the indication of the amount of excess anions in the crop residue. Okay, as indicated here as the minus signs, the negative charged as a organic compounds. So organic anions can protonate to form the acid, while the organic acid can deprotonate to form the organic anion. So both anions, organic anions and acids can physico-chemically react with the functional groups of the soil colloids, inorganic soil colloids. Okay, so this kind of reactions can affect the soil pH. So the effect on the soil pH can be related to the initial soil pH and the pKa values of these organic compounds. If pKa values of these organic compounds are greater than initial soil pH, addition of the crop residue is expected to increase soil pH through the physical-chemical reactions. But more importantly, decomposition of the organic anions is more important to increase or contributing to the pH increase through the decarboxylation process. For the organic acid, if the organic acid is fully decomposed during the decomposition of organic residue, then there is no net effect on soil pH from our own research. So this slide shows some of the results to validate the hypothesis or some of the mechanisms proposed in the previous slide. So in this incubation experiment, we include the 3 soils with the initial pH of 3.9, 4.5, and 5.1. So the soil pH still in the acidic range. This time we add the 5 crop or plant residues, as well as the no residue control. As they illustrate the pH line, the bottom line is no residue addition control. Okay, so we incubate the soil with residue for 105 days. And from here, you can see that addition of the residue increases the soil pH, but to the various degree, depend on the residue type. So for the soil of the initial pH of 3.9 and 4.5, the pH increase is maintained until day 105. But if you look at the soil, your right-hand side of the panel, and the soil with initial pH 5.2, the pH is declined after the initial increase. So this decrease of the pH after the pH peak is related to the nitrification processes. We know nitrification is an acid-producing process. So based on the results, if I can calculate the amount of alkalinity released to the soil based on the pH increment and pH buffer capacity of individual soils, and if I also calculate the amount of alkalinity added as the plant residue minus the alkalinity contributed from the nitrogen cycle, we are able to plot between these 2 figures the alkalinity produced due to the plant residue addition and alkalinity added into the soil as the x-axis. We can see the very nice linear positive relationship between these 2. But if you look at the slope of these regression lines, you will find that the soil with the initial pH of 3.9 had the highest or greatest slope followed by the soil with initial pH 4.5, and the lowest in the soil with initial pH 5.1. So that's verified that the initial pH can affect the alkalinity release from the added crop residues. Now, in another experiment, we added 6 crop residues which have various concentrations of excess cations. As I mentioned, is the indication of the amount of the organic anions in crop residues as excess. Okay, range from 25 to 140 or 35 centimoles per kilo crop residue. Y-axis is alkalinity, amount of alkalinity produced or generated due to addition of these crop residues, and built in the pH increase as well as the pH buffer capacity. So half of the jars, if we fumigate it with chloroform, minimize the biological reactions. So under sterile condition, we can see the still positive linear relationship between two, right? If another half of the jars incubated under normal condition without non-sterile condition, that means there are biological reactions, we found that the increase of the alkalinity amount is much greater. It's about twice more compared to the alkalinity produced under the sterile condition. If we compare the two soils, the water soil with initial pH 3.7 on your left, and the landslip soil with the initial pH 5.1 on your right, and you can see again the slopes of the soil with a lower initial pH are steeper or greater. Okay, so indicates that the initial pH is important. Affecting the pH increase through the pure physical chemical reactions, and as well as the biological reactions, that means decomposition. And decomposition is more important for the pH increase. Non-crop residues, as mentioned, everybody, most of you are aware that the crop residues have the soluble fraction or soluble component and insoluble component like CO1 material. Okay, then we separate the soluble fraction from insoluble fraction of the crop residue. So here is a result from chickpea as an example. Okay, so chickpea has a high concentration of organic anions as indicated as the excess cations. So if we add the soluble fractions into the soil, the Podosol, which have an initial pH 4.4, and the Tenosol of the initial pH 6.2, we can see that adding the soluble fraction can dramatically increase the pH in the initial stage, as indicated in the red lines. Okay, so the increase of pH is much greater compared to the effect of insoluble fraction. Then the pH decreased because of the nitrification process. So then we asked ourselves, whether that kind of pH increase can last longer, can persist over the time, so that the impact on the soil pH in acid soil can be a little bit longer lasting. So we did the long-term field trial. Okay, so the residue was applied in the top 10 centimeter soil column. Okay, we buried these columns in the open paddock field. So all the climatic kind of conditions are same as the field paddock. In this experiment, we again include two soils with initial pH of 6.2 and 4.4. We include 3 crop residues that include wheat straw, which have a very lower concentration of organic anions. Canola residues have a higher organic anion concentration but low nitrogen concentration. And chickpea has a high organic anion concentration as well as a high nitrogen concentration. So this slide shows the part of the result. So I select the Podosol as an example, and with chickpea and chickpea residue and wheat residue as two extremes. Okay, so that's the original pH profile of these columns. They're about 4.5. So when we add the crop residue, we found that the pH increased over 1 unit pH unit, over the 1 unit pH, okay, in the applied layer. That's within 1 day of the application. So the response is very quick, less than 24 hours, right? And then that's the pH profile 2 months after the residue application, 40 months after, and 4 years after. So we still can see the pH increase after addition of the chickpea residue, at least 4 years after initial application. So the pH is slightly decreased over time is because of the reacidification of the soil. And we also can see the pH below the applied layer is also increased. In contrast, if we apply the wheat residue into the soil, there was a little effect on the pH, irrespective of the 1 day or 4 years. So now everybody knows that soil properties can affect plant growth. On the other hand, plant growth or growing plants can affect the soil properties, especially in the interface between root surface and the soil that we are talking about the rhizosphere, okay, as mentioned at the very beginning. When the crop plants grow in the soil, they need to take up nutrients as well as water. At the same time, these plants can release the organic compounds through the roots. So these organic compounds, or we call the root hectares, can affect the microbial activity, composition, or diversity, which in turn can affect the decomposition of soil organic matter. So in the last part of my talk, I will discuss about the rhizosphere effect on the decomposition of soil organic matter. First, we developed a device that can harvest the CO2 produced from the organic matter, soil organic matter decomposition, as well as the CO2 from the root respiration. This time, we use C4 soil, which had been grown with the C4 plants for over 100 years. So because of some physiological mechanisms, the soils with the C4 plant history can relatively enrich the C13 natural abundance. So this natural abundance differs or higher than the natural abundance of CO2 derived from root respiration. So use this C13 natural abundance technique, we are able to separate where the CO2 comes from. So here we look at the plant species effect on the acceleration of organic matter in the soil. So this time we call it rhizosphere priming. From here, we have the 3 legume species: chickpea, field pea, white lupin. So these 3 legumes will rely on the nitrogen fixation. We also grow the wheat plants rely on the urea. Okay. From here, we can see chickpea and field pea have the lower rhizosphere priming than wheat or white lupin. And at days 56 and 84, we also can see the negative value. That means the favors the carbon or the carbon sequestration. Then in the next experiment, we use a different nitrogen form. Okay, nitrate and ammonium both are the available form for plant uptake and exist in the soil. So from here, we can see nitrate nutrition increase the rhizosphere priming, while ammonia doing the opposite way. And the reason is that nitrate can increase the rhizosphere pH, whereas ammonium nutrition decrease the rhizosphere pH, as indicated in the green bar. So pH, rhizosphere pH can affect the microbial growth or the microbial biomass carbon. You can see that microbial biomass carbon was much higher in the nitrate treatment than in the ammonium treatment. And microbial respiration in the rhizosphere also for the same effect. Irrespective of the plant species or nitrogen form, we are able to establish very nice positive relationship between the rhizosphere priming, that means acceleration of the organic matter decomposition due to the plant growth, and the rhizosphere pH. When the rhizosphere pH is lower, below the 4.5, for example, we found there is a negative rhizosphere priming, which favors the organic matter decomposition, organic matter sequestration. So in conclusion, summarize what I've discussed so far is that long-term liming history either decrease or do not affect the concentration of soil organic matter, nor its stability, but the soil comes from the topsoil. And residue decomposition has a higher optimal pH than the acceleration of the soil organic matter decomposition induced by residue application. So let's give you the important implication how we can manage the acid soil in terms of the liming to what kind of target pH. So soil organic carbon decomposition or priming correlated strongly or positively with the abundance of archaeo genes as well as lignin decomposing genes, but negatively with the total carbon after lime application. Decomposition of crop residue increased the soil pH, but this increase was proportional to the concentration of excess cation or organic anions in these residues. And we say the decomposition of these organic anions or decarboxylation was the major cause of the pH increase upon the residue addition. And we demonstrated that the rhizosphere pH or the acidification, that means decreasing the pH, negatively correlated with the rhizosphere priming. So that may favor the carbon sequestration, but no good in terms of the acidification point of view. So future work should cover the wide range of soils under various climatic conditions, various kind of cropping regions, because here we only include the limited number of the soils. And I think it is worth exploring the impact of pH and surface lime application on the dynamics of soil organic matter in the subsoils below the plant layer. I think it's also important or interesting to illustrate the role of the legumes in rotation on soil acidification, that is very well known, and build up the nitrogen fertility and carbon size, that is also known. But what's the combination? Whether the soil acidification or rhizosphere acidification of the legumes contribute to this kind of carbon stability, build up the nitrogen, the organic matter concentration in the farming systems. So finally, I would like to thank many people who contributed the work I reported today. Those includes my former PhD students, my current colleagues, my former colleagues, and my collaborators. So thank you very much for your attention and for your patience. Thank you.
I'm Samantha Grover, and it's my great pleasure on behalf of the Victorian branch of Soil Science Australia to sincerely thank you, Professor Tang, for presenting the 2020 Leeper Lecture. I know that Leeper would be pleased with today's event because of the scientific rigour, but also the clear, simple English communication of the presentation that you've all just heard. Now, we've had a number of questions have come in to the Q&A box already as the talk has been progressing, and Tang is going to have the opportunity to answer some of those questions. But while he catches his breath, I'd like to encourage and remind people how they can ask questions if things have come up during the lecture that they would like some further discussion with Tang around. So some of you will be very familiar with Zoom and perhaps some less so. In the bottom of your screen, in the centre, there should be a Q&A box. And so if you click on that, an option will come up for you to type in a question, and then I'll read those out to Tang and he'll be able to answer them and have some discussion around your topics of interest. If you need further instructions with that, you can type in the chat to get some more instructions, but try and put your questions in the Q&A box and other comments in the chat. Okay. Tang, we've got quite a few questions. Your talk has generated a lot of interest. I'm going to start off with a simple one which was asked right at the beginning by David Rees. So David writes, what method of pH was used?
So in Australia or in our research, we generally use a 0.01 molar calcium chloride in 1:5 ratio and shaking for 1 hour.
Thanks, Tang. I think that covers all the points that David put. The next question is a more a bit of a longer one by Alice Debney. So I'm going to read it out in two parts. The first part, Alice says, are you able to please explain in more detail crop residue behaviour in the carbon cycle?
Okay, so I think that from that carbon cycle side, the effects of the carbon cycle on the pH, so the most dominant kind of mechanism is the decarboxylation or decomposition of the organic anions within the residue. Then about 20 to 30% of that pH increment could be the undecomposable component which directly acts with the functional groups of the soil colloids, like cellulose, lignins, they all have the functional groups. But the effect of that component on the soil pH change depends on the pKa value of these compounds. Is that clear enough? And Alice, we found that the priming effect after residue addition can contribute the slight alkalization effect. That means that can induce a slight pH increase.
Thanks, Tang. Alice also says that she often has colleagues asking her about, particularly around stubble retention. So when you retain stubble from crops, does that not mean that they sequester carbon? And she says she can't find papers or articles around continuously cropped systems that are able to increase soil carbon stocks.
So you are saying that when they add this crop residue, that's not necessary to increase pH, right? But still increase the carbon sequestration. So what I reported here is based on the acid soils. So if it's a moderate acidic soil or the neutral soil, the story may be different. Obviously, return of crop residue is a very important practice to increase the carbon sequestration. At the end, it really depends on the balance between each contributing factors, right? And different crop residues have different impacts on the soil pH. Like cereal crops, they have very little effect on soil pH. Therefore, pH may not necessarily impact on the decomposition of soil organic matter in the long term. But if you look at legume residues, they have the high concentration of organic anions. So when they return the legume residue to the soil, they will increase pH, but followed by decrease of pH because of the nitrification process. But growing legumes will decrease the rhizosphere pH because of cation-anion balance. They extrude the hydrogen ions into the soil. So during the legume growth, and they tend to decrease the pH, especially around the roots in the deep soil layers. But to return back the residue into the soil, they will increase the pH mainly in the topsoil. So it's more complicated. But in the end system, everything should be balanced. If there are no leaching loss or losses through the gases.
Thank you, Tang. As you say, it's a complex, there's lots of different processes going on. And we've got a few more questions here. I think I'm just going to have time to ask one more, which is so this question is specifically referring to a paper, Butterley 2011, and it's from Wongan Hill. And he says, "Hi, Professor Tang, could you explain why the pH decreases are only found in the soluble fraction treatment, but not in the whole residues and insoluble materials in this study, Butterley 2011. So, if nitrification occurred, it should be happening in the whole residues treatment." Does that make sense?
Okay, that is a very good question. So from my understanding, the soluble fraction also contains soluble nitrogen and protein. Okay, so these are the organic nitrogen. During the decomposition of that soluble fraction, the nitrogen is mineralized, then it's a large proportion released to the soil as ammonium. So this is a pH increasing process, but followed by the nitrification process because we induce the pH higher, so that favors the nitrification process. Okay, but in the insoluble fraction, I think that the nitrogen other decomposition is slower, so we may see the gradual decrease over a longer period of time. But with the soluble fraction, the decomposition of some soluble compounds can happen within a day.
Thank you, Tang. There are more than 180 people have joined your Leeper Lecture, which is a record for Soil Science Australia Leeper Lecture. Thank you very much. There are more than a dozen unanswered questions. So your talk has generated an enormous amount of interest. It is the end of our hour. So I'd like to thank Professor Tang, and lots of people are wanting to do a Zoom clap and writing in the chat about how they can thank you. But I'd also like to thank everybody who's joined, both people who would regularly attend the Leeper Lecture in person and those of you who are joining for the first time in this online format.
Thank you very much for having me, and thank you for all the audience for your time. Thank you.